Okay, hi everyone, welcome to this week's Great River Rapport workshop. We are currently here at Morrisburg Beach joined with the Finns crew, and we are going to be walking through all the different things that the Finns crew works on. So from drone surveys, eDNA sampling, water quality testing, fish seining, and plant cover surveys. We'll be going through every step that the Finns crew undertakes. And we are also joined today with Future Climate Leaders, so with Lee Theodore from Future Climate Leaders, and we will be sort of taking on a climate change lens, investigating the ways in which climate change is impacting certain aspects of the research conducted by the Finns crew. And we are also happy to be joined with Dr. Brian Hickey, who will be sort of the commentator for the workshop today and explaining the processes happening. So I will pass it on over to Brian.
All right, well, hi everyone. If there are some people joining us live, we got off to a little bit of a late start, and that's all in a day's work for anybody doing field work. We visited a site earlier that used to contain water, and now the water is gone. It's completely dry, filled in with vegetation. And as climate changes and as water levels get more and more unpredictable, perhaps that's going to happen more and more regularly. Anyway, we moved to lovely Morrisburg Beach, where there is water and where we're going to go through all of those things Yannick mentioned. And I'll start first with explaining what FINS is. FINS stands for Fish Identification Nearshore Survey, and this project, which has been led by Matt Windle, one of our research scientists, has been looking at the nearshore fish community, the small minnows, the fish that are sometimes by management biologists lumped under the derogatory term forage fish, because the only important reason for them is to be forage for the bigger fish that anglers want to catch. But as a biologist, they're the really interesting fish in the river because they're the most numerous. There are many different species. They're being impacted by climate change. They're being impacted by the introductions of invasive species like round gobies, and we want to understand that fish community because that's the fish that supports the large sport fish that most people care about and most anglers like to go and look for when they're angling. So one of the first things we're going to do is talk about drone surveys. So this is a component. This project's been going on since 2015. In the last couple of years, Matt Windle and his crew has included drone surveys, and you can see our drone here. This will give us high resolution imagery, an aerial view of the sites that we're sampling for fish and water quality and plant cover, and it'll allow us to track changes over time with much better resolution than we've been able to do in the past. And so Evan, I think, is ready to launch this machine, so I'll get out of his way.
Take off.
And so as part of this project, Matt has more than 50 different sites that span all the way from Lake Ontario in Prince Edward County to the area near Montreal, Quebec. And so this is one of the most extensive surveys, I think, ever, both the most extensive and intensive, because his crew is out sampling these sites almost every day during the summer, or at least several days each week. They have a couple of days for planning and maintenance activities, but they're out here 3 days out of 5 every week throughout the entire summer, and as you can see today, right into the fall. We're just about wrapping up for this season, but in total, Matt's caught more than 155,000 fish, all of which, or most of which, have been identified and then released back into the water. So one of the other components of this project is understanding changes in water quality, and once again, as climate change happens, water quality changes. And one of the underappreciated effects of climate change, as climate warms and as we have in some water bodies lower volumes of water, that means the contaminants that are in the water get more concentrated. And so one of the things we do at each of our sites is measure basic water quality. We measure dissolved oxygen, pH, and several other different water quality measurements, so that we have a record of how things vary among sites in different parts of the river, but also how things are going to change over time as we experience this unprecedented environmental change that's brought upon us by global climate change. So the drone is humming above us, and we've got a couple of people out in the water taking water samples.
Ready, yeah.
Temperature is 21.4.
Okay.
Dissolved oxygen 131.1.
Mm-hmm.
And 11.58.
Okay.
Chlorophyll 0.81.
Okay.
PC negative 0.10.
Okay.
And you still need SPC, right?
Yep.
SPC is 306.5.
Perfect. Yeah, so as Brian was mentioning, I'm Matt Windle. I'm a research scientist at the River Institute as well, and we're in the process right now of just measuring some standard water quality measurements, and that includes temperature, dissolved oxygen, which is really important for fish. They need to breathe oxygen underwater, so we measure the concentration of that in the water. We also look at other variables. We're also measuring connectivity, which has an effect on what species can live in certain types of water. And we're also measuring chlorophyll concentration. So there is some chlorophyll in the water today, which is an indication that there's algal concentrations in the water. And people are concerned about algal blooms. It is food source for a lot of the base of the food chain as well. So it's a really important variable to measure as well. So we also collected, I don't know if you saw Courtney collecting some bottles there. Those are nutrient samples for phosphorus and nitrates. So those are going to be used to give us a sense of how many nutrients are in the water right now. And of course, nutrients feed the food chain, feed productivity. So that's another important variable we look at as well. And that does vary across different types of habitats. So we completed our drone survey here. We've captured overhead imagery of our site, and we do that survey before we actually get in the water to do our fish collections, so that we don't disturb the sediments and we can see the bottom really, really clearly. We also do our water collection and water quality measurements before we get in the water to do the fishing, for the same reason, just to make sure we don't disturb the sample before we take it. So we've done those two things. Now we're ready actually to start setting up for our fishing. The next element of the survey that we can actually show you, though, is the filtrations for environmental DNA. So I don't know, Brian, if you wanted to go show people, or I can go there.
The environmental DNA?
Yeah.
You can go ahead, and then
Okay.
I'll answer. While you're heading over there, I'll answer our first question. We got a question about the mowing that's been happening in Cornwall, right down to the banks of the Cornwall Canal, and whether we have any sites near there. We do have sites in the Cornwall area, though not specifically in the canal. And I think even had we had some sites right in the canal, I think the kind of impact of that mowing is not the sort of thing that's likely to be picked up by this type of a survey. This type of survey where we're monitoring changes in the fish community, that's going to detect kind of chronic things that happen over long periods of time and very slowly. To detect any effects of that mowing, we would have not only had to have had sites right at that spot, but we probably also would have had to have done the sampling right during or shortly after that event.
So now we will be going over to the eDNA sampling and water quality testing.
So eDNA is environmental DNA, and one of the techniques that's being developed now is the ability, rather than catching fish, understanding what fish are living in a particular area by simply taking a water sample and doing DNA analysis to look for fragments of DNA that come from specific species. And right now, this technique is just in the development stage, and we're contributing to that development by comparing the picture we get of the fish community if we simply take a water sample and look for fragments of DNA in the water compared to if we actually catch the fish. I suspect what we're going to find is the two approaches are complementary. Environmental DNA may pick up detections for species that are hard to catch with nets, and vice versa. But we're just in the process, as I say, as a fish research community of developing those techniques. So we're working with a variety of groups. This entire FINS partnership is a partnership with the Mohawks of Akwesasne, but in particular the DNA component is a partnership with some people at Queen's University who are experts in this new technology and on the forefront of developing the technology. So what we do is we filter a large volume of water. You can see our pumps here on the back of the truck, and we'll filter a large volume of water. That will go back to the lab where we'll look for DNA fragments of fish that we may not necessarily catch in the net, but have been hanging out here in this stretch of water and have been leaving traces in the way of fragments of DNA. So we get these fragments of DNA in the water for a variety of reasons. All right, so I might have been interrupted by a technological glitch in our streaming, so I'll just repeat a little bit of my last section there. So what we're doing is we're looking, after filtering a large amount of water through a very fine filter paper. That filter retains little fragments of biological material, scales, mucus, epidermal cells, skin cells, little bits of digested fish that have passed through the gut of a predator fish, cells that have been scraped off the digestive tract of the predator fish during the digestive process. All of that ends up in the water in very, very small, minute quantities. So we filter large amounts of water, get those DNA samples onto a filter paper. We bring that back to the lab, and that's where it undergoes the genetic analysis. And the more we learn about genetics, the more we are able to develop markers, sections of DNA that we know are diagnostic of particular species of fish. It's a very similar technology to the technology that gets used on CSI, on TV to track down perpetrators of crime, except that we don't use those blue lights and glasses, which is all for show. Hate to break that news to you that what you see on TV is not real science.
So as you can see on this paper here, this is the filter that picks up all of the DNA that's running through the water. So the pump is pushing the water through the filter paper, and it's picking up whatever signals it can. And you can see, here I'll just fold it, you want to fold it kind of so that the eDNA is on the inside. And then when I hold it up, you can see the fragments of DNA on the filter paper.
And there'll be DNA from a lot of different things. There'll be algae, there'll be plant material, and in the lab we will expose this to particular probes that look specifically for the fish species that could potentially be in this part of the river. And how much water are you filtering through that little piece of filter paper?
So depending on how turbid the water is, It could take a few filters per bottle, but the bottles themselves are 1 liter.
Okay.
And we're doing 3 1-liter bottles at different areas along the section that we're seining for fish. So we're able to find a couple different areas and waypoint it, so we'll have the coordinates and be able to see exactly where we found that signal in the water.
Okay, great. And so over the as this data set develops, Matt and his crew will be able to look at the picture of the fish community we get, looking at traditional fish seining methods, using a net to capture and corral fish and identify them that way, compared to what we get using this new eDNA high-tech approach. And so next, I think we'll, should we head down to see whether they've started seining yet? I'm guessing they should be underway now. If not, they will be at that shortly. So obviously a key component, since we're studying fish, is to capture fish and see what species we have here. And you can see when we get there in a second, you can see them dragging a large net through the water. That's called a seine net, and they're simply corralling the fish that are in the water. You can see a couple of people in advance of the net kind of flushing, scaring fish into the path of the net, and we have a couple of people on the on each end of the net that dragging it through the water. They'll come to shore, and the net will have corralled a bunch of fish, hopefully, and then we'll transfer them into buckets of water, identify them, and then in most cases we release them all back into the water. So we'll head over a little closer to them and see if we can get a look and see what exactly they've caught.
Is this? So Courtney, what is this called?
This?
Water taxi?
Water taxi.
Fish taxi.
Yeah.
So you can see we've just completed a single survey. We're going to do 3 of these surveys as part of our overall, or 3 hauls as part of our overall survey. Each haul is 20 meters along shore, so we do the same length of haul so that we can compare effort across sites, and we make sure we put the same amount of effort to catch the fish, so then we can compare how much fish we caught between each site. And that makes sense, right? If you did 1 haul at one site and you did 5 at another, you would expect to catch more fish at the 5 haul sites. So we make sure we only do 3 hauls per site and the same length of shoreline, just to standardize that. The team right now is just collecting some of the fish out of the net here, and you can see some different species, some brook silversides, for example, those long fish there. This is a fairly sandy, rocky habitat for fish. There's not a lot of vegetation, so we're not expecting to find a lot of fish abundance or fish diversity just because that lack of vegetation. The vegetation provides more structure, more places for fish to hide and live. But we might be surprised. We're not sure. We'll see how it goes. Today, we're definitely probably going to catch a lot of invasive round gobies here, because we've seen them all over the bottom as we were doing the survey. So you can see in our fish taxi here, we have our first catch. So we're going to repeat this now another time.
And so the biggest thing when you're kind of holding fish like this before measuring them is you want to reduce their stress as much as possible. So we have some aeration pumps that Kate's going to go grab, and it's going to increase the oxygen level. There's holes in the side of the water taxi to kind of bring fresh water in, and so we want to keep the fish at a good temperature and oxygen level while we handle them.
we actually have a follow-up comment from the previous inquiry about waterfront properties.
Yep.
It says, thanks, it might be interesting to have some test sites right in neighborhoods where you could have direct contact with private property owners who are removing vegetation down to water edge of wetlands and creeks, even when it is on city property. It might help people understand the effects they are having on water quality. It's not so much of a question. But
Yeah, so that's a great comment, and because because I'm not a core part of this Fins crew, I don't know where all of Matt's sites are. But when he's not dragging a net through the water, I'll ask him and I'll revisit that question. I 100% agree. It's possible that Matt actually has some of that information and has some sites in places like that. Sometimes the people who are doing that aren't anxious to have us do sampling on their properties, and that creates a little bit of a challenge getting sites like that. But the point you made, where in some cases they're mowing down to the water's edge even though it's not actually their property, in which case land ownership is not an issue. So yeah, that's a great point. I think the information we'd really want for that kind of a site is less about the fish and more about the water quality, which is a component of this research, but we're not measuring some of the really key things that might be important in the scenario you're describing, like pesticide runoff and fertilizer runoff.
All right. So are there any more things that the Fins crew is doing that we should be covering?
I think we've seen most of the activities that are typical, other than maybe seeing what species of fish they actually caught, which may or may not happen. Too late for us to talk about. But so some of the typical things I've fished for other research projects in this area, we could certainly expect to find some rock bass. Matt talked about how the diversity might be a little bit lower because of the lack of vegetation. There are some fish, however, that like that lack of vegetation. So although we get fewer species, we sometimes get fish that you don't get in more well-vegetated areas. One that I'm particularly interested in is the Cutlet Minnow, which is a species that's quite rare in the St. Lawrence River, and it's only found in this part of Canada, so it's unique to Quebec and Ontario and the St. Lawrence and Ottawa River drainage basins, and that's a fish that actually requires rock and not too much vegetation. So I'm hoping they catch one of those. That species is known from this area, although it's much more abundant near Cornwall. Matt, one of the questions we got was, do you have any sites where people are often clearing right down to the shoreline, often on property that's not actually their property, it's city property, but they mow right down to the river in the immediate Cornwall area? Do you have any sites at places like that?
I'd have to check. We have some older sites. We did a really big effort in collaboration with the Mohawks of Akwesasne back in 2015 and 2016. A lot of sites around Cornwall, Lake St. Francis. We did a lot of shorelines around there, so I'm sure we probably captured some sites that have that feature with people coming down. That's a good question, because that's something we're interested in. Is the fish habitat impacted by people mowing right to the edge of the shoreline, instead of leaving a nice riparian zone of vegetation along the shoreline and simulating a more natural habitat for the fish, a more natural shoreline habitat? When people leave trees and brush along the shoreline, it creates shade and canopy cover, which then can actually cool the water a little bit and offer little places for fish to find refuge from the heat or predators. So that's a really good question, and for sure we've sampled some sites like that around Cornwall. I can talk a little bit about the fish aspect as well.
Great if you've got fish there. Yeah.
Yep. So we've as part of this survey, I think Brian, you probably mentioned the effort we put in. We have over 200 sites in the St. Lawrence River, from Montreal all the way down to Prince Edward County. 200 sites we've sampled, and we've documented over 155,000 fish over the past 7 years as part of that survey effort. So we have a really good idea now of the baseline fish communities of the nearshore St. Lawrence River, and that covers wetland habitats, rocky shorelines like this one, developed shorelines next to homeowners, and then really wild landscapes as well. So part of this effort too is documenting the distribution of species at risk. And there's a handful of species in the St. Lawrence River that are either endangered or they're threatened, and there's always some knowledge gaps where we need to fill in. Where are they located? What kind of habitats do they like? Are they doing okay? Are there certain areas where we would expect to find them but we don't, and why not? So some of the fish that we're interested in are minnow species. That people probably just group all together as shiners, but there's dozens of different minnow species that live in the St. Lawrence River, and one of them in particular we're focused on is the pug-nosed shiner, which has it's a little tiny minnow with a black stripe across its body, very small face on it, small mouth on it, as well as other species of risk. We're documenting those as part of the survey. We also document the movement and increasing distributions of invasive species such as round gobies. There's a new species in the river, a tube-nosed goby, that's moving downstream from Kingston this year.
We're back.
Okay, so we have in this bin here, we mentioned before that when we catch our different hauls, we put them in separate bins just to separate out the catch, and then we make sure we have some aeration. This is what this is doing, just to provide oxygen for the fish to breathe, to minimize their stress. The team's going to come back afterwards and we're going to sort through this catch. We're going to identify all the species we caught, count how many of each species we caught. We're going to measure their lengths as well. And after we're done all that, we put them back in the river. And we can have a quick look to see what kind of species we have in here. I don't have a net handy, but I believe there was some. Okay, so this species is a round goby. So this is from the Pondo-Caspian area, the Black Sea area of Europe and Asia, and it came across in the ballast water of ships back in the 1980s, and it was released into the Great Lakes system back in the late 1980s, and it spread out through the Great Lakes system and is now found in all the Great Lakes. It covers these individuals. Sure. So one of the questions we just had was, what do we do with the round gobies once we catch them? And that's a great question. It is an invasive species. Technically, some agencies of government are required to remove them from the river when they catch them. This species is probably the most abundant fish now in the Great Lakes by numbers, and certainly in the St. Lawrence River by numbers. It covers the bottom. So for us to remove a handful accomplishes nothing. It's a drop in the ocean, essentially. So we end up just putting all of our catch back in the river. It accomplishes nothing to sacrifice or kill the fish, so it's not really part of what we're trying to accomplish with this survey. There are other species of invasive fish, however, if we do come across them, that we do collect and keep and we don't put back in the river, and those are new species where they haven't really established themselves yet in the St. Lawrence River. So by keeping them back, we can actually potentially reduce their numbers or distribution much more so than we could with round gobies, which are super abundant at this point. So good question though. If people do use them as bait or other things, I mean, you're technically not supposed to move them around if you're using them as bait. So if you do dispose of them, make sure you don't move them between different water bodies, because they're able to survive and reproduce quite easily that way. So I don't know if you want to head back to the shore now and back to Brian.
Okay. So we'll let Brian just there anything else you'd like to add, Brian, before we close it
So
off?
I just want to come back to that question about the clearing to the shoreline, and do we have any sample sites there? Because I want to make sure I'm not sounding a little bit wishy-washy on that, as some scientists tend to do. Science is not good at pinning one little tiny event in time and space on a change in an ecological community. So yes, we could move sampling sites to those locations, but we don't need sampling sites at those locations to know that clearing land to the water's edge is a bad idea. There's lots of science that's been done for lots of years that tells us that that is a bad idea. It doesn't take a sampling site to tell that spending gasoline and dollars and spewing out more climate warming gases just to cut some vegetation is a bad idea. So we don't need a sampling site to draw the conclusion as a scientist that that's a bad idea. So I just want to be very clear about my opinion on that whole issue. And Matt may or may not have sampling sites. I'm not going to wait for his data to conclude that that's not a thing we need to be doing.
All right, so we'll end it here unless there are any other questions. I'll just give a brief look at the comment section and see if there are any other questions that have come in. It doesn't look like it. So we're going to end it there for today. So a big thank you to the Fins crew, of course, for all of their work and for allowing us to join in with them today. A big thank you to Dr. Brian Hickey for giving us so much knowledge on everything that's going on. We'd like to thank everyone for watching, for asking all of your questions. And this is one of our Great River Rapport workshops, which is a series of Great River Rapport workshops. If you'd be interested in knowing more about the River Rapport itself or any other workshops we will be conducting, please visit riverrapport.ca/workshops. All of our past workshops and upcoming ones will be posted there. So we will continue to be hosting the series for the next few months, and we look forward to having you join us. Thanks.